So hello everyone, sorry for the technical inconvenience. We have technical issues with the transmission, so this is the new transmission of this webinar. Thank you very much for your patience. So remember that this webinar is recorded on the YouTube link that I previously sent to you. So thank you very much, and Maria Andrea, please go ahead again. Thank you.
Okay, sorry guys for the technical difficulties, but okay. So I'm going to talk today about the gamma delta T cells and the blood stage human malaria, which is the work I've been doing for the past 3 years during my PhD. So malaria is a mosquito-borne disease caused by the infection with one of the 5 species of the genus Plasmodium. The malaria parasite cycle is complex and involves 2 hosts, the mosquito and the human. And it starts when during a blood meal, the Anopheles mosquito injects the sporozoites into the skin. These sporozoites travels and reach the liver, infects the cells, and then mature, rupture, and release thousands of merozoites, which are the ones that infect the red blood cells. Then within the erythrocytes, these merozoites also mature and rupture and release again thousands of merozoites, which infect new red blood cells. And some of these, they differentiate into the gametocytes, which then are taken up by a new mosquito during a blood meal. So it's during this blood stage where the parasite releases toxic factors that contribute to inflammation, and the complications of the disease arise during this stage, which are characterized by symptoms such as severe anemia, respiratory distress, metabolic acidosis, and the most fatal, cerebral malaria. Therefore, to officially prevent severe malaria, tight control of the parasitemia is essential. Malaria depends mainly on climatic factors such as temperature, humidity, and rainfall. Malaria is also transmitted in tropical and subtropical areas where the Anopheles mosquito can survive and multiply. Because I told you that the temperature is particularly critical, generally in warmer regions that are closer to the equator, the transmission is more intense, and also the malaria is transmitted all year long. The highest transmission is found in Africa south of the Sahara and also in parts of Oceania. As I told you, temperature is critical. So, for example, the species that causes severe malaria, which is Plasmodium falciparum, if it's below 20 degrees Celsius, it cannot complete its growth cycle in the mosquito, and therefore it cannot be transmitted. But there is another species that is Plasmodium vivax, which in cooler regions might be more prevalent because it's more tolerant to lower temperatures. Protective immunity responses to malaria have been shown to be highly complex and requires the fine-tuned interplay of innate and adaptive mechanisms. So in the case of the immune response, just to remind you, the innate immune system is primitive, is rapid response, does not require the presentation of an antigen, and does not lead to immunological memory. The effector cells are the neutrophils, the macrophages, and the mast cell, which they react within minutes to hours with the help of complement activation and also cytokines. In the case of the adaptive immunity, a slow response is provided by the lymphocytes, which recognize unique antigens through cell surface receptors. You have, within the adaptive immunity, you have the B cells, which constitute 20% of the blood lymphocytes, and they are used in the production of antibodies in fighting bacteria, viruses, and tumor cells. In the case of the T cells, they comprise 80% of the blood lymphocytes, and they are used in identifying antigens and in releasing chemicals which attract macrophages. And the T cells, they destroyed virus-infected cells, foreign tissue, and tumor cells. In the case of malaria, the role of antibodies is been of great interest And it's been shown that these antibodies can inhibit the parasite growth at different levels. For example, they can inhibit invasion of the merozoites in the red blood cells. They can also eliminate free merozoites, or they can eliminate infected red blood cells by phagocytosis and complement activation. However, less is known about the cell-mediated response during the blood stage. There has been shown that there is a particular subset of the T lymphocytes who bear the gamma delta T cell receptor, and it has been demonstrated to be important in defending the host against a broad range of infections. This is achieved through 6 main mechanisms. So these gamma delta, they can produce diversified set of cytokines and chemokines to regulate other immune and non-immune cells. They can also provide help for the B cells and promote the production of IgE. They can present antigen for alpha-beta T cell priming to trigger dendritic cell maturation, and they can regulate stromal cell function via growth factor production. But the most important role is the lysis or strep cells. Additionally, it has been shown that when patients are infected with Plasmodium falciparum malaria, there is an association with an increase in the percentage and absolute numbers of these gamma delta, as you can see here. But still, the function of these cells is still not fully understood. As I mentioned before, there is no information about the role of the cytotoxic T lymphocytes, and also there is no information about the pore-forming proteins that are derived of these cytotoxic T lymphocytes and their action on defected red blood cells. Among these pore-forming proteins, they are the perforin, which its activity is dependent on cholesterol content, disrupts cholesterol-poor membranes of bacteria, fungal, and parasites. And there is the serine protease, the granzyme B, which is secreted referring to mediate the apoptosis in target cells. Recently, it has been shown that these cytotoxic lymphocytes kill intracellular bacteria and certain lymphocytes such as Trypanosoma cruzi and Toxoplasma gondii. In the case of bacteria, they actually they conclude that the granulysin introduces the granzymes into the bacteria, and there inside the bacteria, the granzymes attack the electron transport chain complex and also some enzymes of the oxidative defense, inflicting irreparable damage in the bacteria. In the case of unicellular parasites, for example, Trypanosoma cruzi, they conclude that it's perforin who delivers or which delivers granzyme into the infected cells, and then granulysin delivers granzyme into the parasites. And there, the granzyme again generates superoxide and inactivate oxidative defenses to kill the parasite. So we follow up on this line of research and address the question of how gamma delta T cells restrict the growth of blood reciting Plasmodium falciparum. So this question was answered in a paper that we published at the beginning of this year in January. And here we found that when we co-incubate the gamma delta T cells with parasite supernatant, that resulted in a massive expansion from less than 2% to over 60% after 14 days. So here we have 3 different conditions. So we have only IL-2. You could see that we only have 2% of gamma delta. When we have the gamma delta with the supernatant of only erythrocytes plus IL-2, you see that we only got 16% of the gamma delta. But when we have the gamma delta with the parasite supernatant and IL-2, you see that we have 60% of the gamma delta were expanded. Then also these cells that were activated with the parasite supernatant, they grow in big clusters, as you can see here, and most of which highly express the gamma delta T cell receptor, as you can see here in green, while less actually showed the CD56, which is a marker for NK cells. Also, when we use high resolution confocal microscopy, we saw that these gamma delta T cells form conjugated with both the early stage of the parasite and with the late stage of the parasite. So you can see here the gamma delta are in green, and here are the parasites. And also within many of these conjugates, the granzyme B, which is in red, it was transferred to the parasite. So here is again a gamma delta. You see here a parasite. Then you see that when the gamma delta becomes in contact with the parasite, you see that there is transfer of the red dye, which is in this case granzyme B. And then also this granzyme B is found in close proximity of the parasite DNA. Then in addition to this transfer of granzyme B into the infected red blood cells, we also saw that there was a reduction in the viability of these parasites. And we saw that by using the mitochondrial potential indicator JC1. Just shortly, if the mitochondrial potential is normal, the dye will aggregate and will display a bright red signal. If the mitochondrial potential is disrupted, then of course it's going to be less aggregates of the dye, and therefore the red signal will be faint. And if the mitochondrial potential is absent, well, there is not going to be any aggregate, and of course we don't see any red. So here we have again a gamma delta. You see here a very happy parasite, which is really a red signal. And then as soon as the gamma delta start coming in contact with the parasite, you see that the red signal start to fade until when the conjugate is formed. Here in this panel, you see that there is no red signal. So here we said that these killer cell infected red blood cell conjugate reduce the viability of the parasite. Also, we wanted to test about the reinvasion capacity. So what we did is that we co-incubate the gamma delta T cells and the parasites, and we have different effector target ratio. And as you can see here in this part of the graph, you see that the gamma delta T cells reduce the reinvasion capacity of the parasite. And then also we did a pretreatment using inhibitors for granzyme, the DCI, and you see that here the DCI completely abrogated their ability to block reinvasion. So here, these results actually tell us that the gamma delta reduce the Plasmodium falciparum viability and the reinvasion capacity, and that actually this is dependent on granzyme. Then we wanted then to dissect the molecular events of this granzyme B-mediated killing. So we tracked the delivery of labeled granzyme into the infected, into the red blood cells. So we have three conditions. We have red blood cells, so not infected erythrocytes. We have infected erythrocytes with the early stages of the parasite, and we have also with the latest stages. So we treated either with perforin plus the green label granzyme B, or with granulysin plus the green label granzyme B. And we saw that in the case of perforin, it delivers the granzyme B in the red blood cells, so in the normal erythrocytes, not infected. In the early stages, you could also see green, and also in the later stages, but it's unspecifically delivered. In the case of granulysin, you see that we don't see any green in not infected, also not in early, but we see mostly in early stages. So if you remember when I give you the introduction, I told you that perforin, its activity is dependent on cholesterol, and in the case of granulysin, it attacks more the membranes that are low in cholesterol. So also what is being shown or has been told is that when the parasite is growing within the erythrocytes, also it depletes the erythrocyte of cholesterol. And then probably that is why we see that perforin is more into the red blood cells and in the early stages, which are they have more cholesterol, while the granulysin goes more into the later stages, which is depleted of cholesterol. Additionally, we also observe a drastic ultrastructural morphological alteration in transmission electron microscopy. So we have here, we have untreated, so infected erythrocytes, but they're untreated. You see here, you can see the apical organelles, you can see the nucleus of the parasite, you can see also the membrane of the red blood cell. Then when we treat it with granulysin, you see that there is no difference between these two. You could still see all the different organelles, But when we treat it together with granulysin and granzyme B, then you see that there is a drastic alteration in the morphology of the parasite. There is loss of hemoglobin, and even there is almost a complete loss of all the organelle features. So until now, all these were done in vitro. So we see that the gamma delta expanded when they are co-incubated with the parasite supernatant. We see also that they form conjugates with the parasite. We see that they transfer granzyme B. And in the case of early stages, it requires the help of perforin. And in the case of later stages, we see that granzyme B is transferred with the help of granulysin. So now we wanted actually to investigate the role of this granulysin in vivo. So one thing is that the rodents, the mice, they don't have granulysin. So we actually have the advantage of a mouse model transgenically expressing the human granulysin. It's in the bulb C background. And we decided also to start with one of the species that infects rodents, which is Plasmodium joelliae. We have this GFP P. joelliae stabilite that we got it from France. And in the case of P. joelliae with the bulb Cs, it causes anemia and hyperparasitemia. So we infect 5 females wild type, which means that they don't have the granulysin, and 5 females that have the granulysin. And then I track the GFP using flow cytometry. So here we saw that already at day 5, you see like around 6% of parasitemia in the blood, of parasites in the blood, and there was a peak around day 7. There is around 16, but you'd see that there is no difference between both groups. And then around day 12, you see already that the parasitemia, or it's no longer possible to see parasites in the blood. So basically that the animals cleared infection. So then we decided to repeat the experiment. So we infect again 5 animals, 5 females wild type, 5 females granulysin transgenic mice, and also I decided to reinfect these animals. And then I see exactly the same pattern, that is around day 7, or in this case around day 9, that there was a peak in the parasitemia. So you see around 20% of the parasites, but again it declined until after day 12 or day 14. I don't see any parasites anymore in the blood. But in the case of the ones that I'd reinfect, as you can see here, the blue and the red, you don't see any parasites, or I didn't see any parasites during the whole experiment. Then when I was start looking or researching about this particular strain, I found out that this is a virulent strain of Plasmodium joelliae, and it actually produced a self-limited infection. And finally, at the end, the animals cleared the infection. And in addition, also, this infection with the non-lethal P. joelliae causes a change in the immunological status of the host and makes it not only clear the infection, but also they become successfully resistant to reinfection. So this particular strain, or this particular strain of Plasmodium joelliae, is not good for to investigate the role of granulysin in vivo. So we check for another one, which is Plasmodium vergii anca. Again, it infects the bulb C mice, it causes anemia and hyperparasitemia. And we got this strain, also GFP tag, from the Netherlands. So again, I have 5 animals, female wild type and female granulysin transgenic. And then here already you can see that there is the granulysin animals actually got higher parasites in the blood already at day 8, and already at day 14 you see that there are like 60% of parasitemia in the blood. So here we see something completely different that we saw on in vitro. As you remember, in vitro the granzyme B actually helps they reduce the viability of the parasite, reduce the invasion, actually is helped by the granulysin or the perforin. But here we don't see that. We actually see that somehow these animals that have the gene, the transgene, somehow they have higher parasitemia. So here, one thing is that the animals that have been chosen for these experiments, they actually have the gene. We are sure that they have the gene. We use PCR to check that, and I only use the ones that are positive for the PCR. But maybe somehow these animals, probably the level of expression of this gene is not high, and that's why we don't see any role of this granulysin. So actually right now I'm doing that. I'm trying to determine the level of expression of the granulysin in these animals. So the coming experiments will be to determine that. And then just to summarize this webinar, so the gamma delta T cells, they bind to Plasmodium falciparum infected red blood cells and release the granzyme B. These gamma delta T cells reduce the Plasmodium falciparum viability and in the infected red blood cells. And the transfer of granzyme B is mediated by granulysin in the case of later stages, and is mediated by perforin in the early stages. And regarding the role of the granulysin in vivo, we still need work to do. Because with P. joellia, we didn't see any difference between the groups, and also they cleared the infection, so it's not good for us or for these particular experiments. And in the case of P. bianca, we see the opposite effect. So as I told you, right now we're focusing also in the rodent to find the expression of the granulysin, but also we are focusing in this part of the cholesterol to understand more about the role of perforin in the cholesterol and also of granulysin. And with that, I would like to thank you for your attention.
So, well, thank you very much, Maria Andrea. I believe that is a very good webinar. This is the first webinar given in English. So we believe that is a very good experience for the program. So now, I believe that we can go to the questions in Spanish. Do you think so? Okay, so...
Yes.
Ok, bueno, nuevamente muchas gracias. Creemos que fue un muy buen seminario. Entonces, recuerden a las personas que están asistiendo a este seminario que las preguntas las pueden hacer en español y las pueden hacer a través del chat de YouTube. Pero antes de que empecemos con las preguntas, yo quisiera que tú nos contaras un poco la experiencia profesional que has tenido como microbióloga, qué has hecho, en dónde has trabajado, con qué microorganismos has trabajado, qué es lo que has hecho desde que te graduaste.
Bueno, yo primero pues cuando estudié microbiología, yo estuve trabajando en la parte forense a nivel de las moscas, pero cuando empecé el doctorado yo he estado trabajando principalmente con parásitos. Son unos microorganismos que me llaman muchísimo la atención, que vale la pena estudiar. Entonces he trabajado con Toxoplasma gondii y los últimos años he trabajado con malaria. Primero trabajé con malaria en ratones, era básicamente la parte del hígado, la infección. Y ahorita he estado trabajando más en la parte del estadio de la sangre y también un poco, como ustedes vieron al final, la parte también obviamente in vivo con los ratones. Entonces he estado trabajando más con malaria, me he enfocado en los últimos 3 años haciendo el doctorado, y hice un internship en Alemania donde también trabajé con malaria.
Ok, bueno, una pregunta que de pronto siempre me hacen los estudiantes que están empezando la carrera. ¿Fue muy difícil conseguir ese paso a Europa? ¿O tú qué le recomendarías a los estudiantes, los que se quieran ir a Europa o a Estados Unidos o a cualquier parte del mundo a seguir estudiando? ¿Tú qué les recomendarías?
No es que, o sea, yo no digo que sea imposible ni que sea difícil, simplemente toca es escribir bastante correos a los profesores. Una cosa que yo he visto es de pronto tratar de buscar un sitio que de pronto con un profesor que tenga financiación, eso puede ser, o por lo menos que te pueda ayudar con la mitad de la financiación, pero principalmente, o sea, es escribir y te toca es escribir, o sea, si a ti te interesa, si ves un artículo y te llama la atención, empieza a escribirle al profesor, o sea, a veces se demoran bastante en responder o a veces ni siquiera responden, pero entre más escribas, en serio que al final te sale algo. O sea, eso fue lo que yo hice cuando yo estaba buscando mi doctorado, escribí a Londres, escribí a Suecia, a Alemania, o sea, escribí yo creo que a todas partes, a Estados Unidos, hasta que al final, digamos, salió primero salió, o sea, una parte de Alemania, que fue solamente para hacer un internship y ya estando ahí en Alemania fue que también me pude contactar aquí con Suiza, con la Universidad de Friburgo. Entonces, todos son contactos y si de pronto sale, si pueden hacer de pronto pósters o si hay algún tipo de, ¿cómo se llama?, de simposio, aprovechenlo también. Y en ese momento es donde ustedes pueden hacer los contactos, o sea, todo lo que sea eso de simposios, conferencias que ustedes puedan ir, así sea simplemente como sea, solamente ir a escuchar, empiecen a hacer contactos, hablen con la gente y así pueden tener más adelante, o sea, ustedes llegan, les dicen que hasta ahora están empezando, que les interesa tal, no sé, de pronto parásitos o virus o bacterias o cualquier microorganismo y, y así se mantiene uno en contacto.
Ok, perfecto. Bueno, tenemos una pregunta. La profesora Lucy pregunta que cómo se podrían aplicar estos hallazgos a la infección en humanos.
Bueno, eso es lo que estamos nosotros tratando de mirar, o sea, con las gamma delta, ¿no? O sea, nosotros sabemos que las gamma delta, como yo les dije, hay una expansión durante la enfermedad, ¿sí? Pero todavía eso es lo que nos falta mirar, o sea, porque si sabemos que la gamma delta es la que de alguna manera transfiere esa granzima y baja los parásitos, o sea, digamos lo que se podría hacer es hacer alguna terapia a nivel celular, o sea, alguna terapia con las gamma delta. Sí, esas son obviamente, como demostramos nosotros en el artículo, que son las que ayudan, digamos, a reducir el parásito, la parte de la viabilidad. Pero todavía nos falta bastante, o sea, comprender un poco más porque también hay otros, o sea, hay otras artículos, se pueden decir, otras investigaciones donde hacen énfasis es en las natural killer, ¿no? Pero nosotros vimos que también son las gamma delta, entonces no sé si también hacer como trabajar con alguna de esas dos, especialmente las gamma delta para mirar a ver cómo se podría también reducir.
Ok, perfecto. Vamos a ver si de pronto tienen más preguntas. Digamos, por ejemplo, una persona que ya terminó el doctorado, digamos, ¿qué tendría pensado para hacer a continuación? ¿Tú qué piensas que harías a continuación después de terminar el doctorado?
Pues una cosa que hacen es de pronto seguir con un postdoc.
Ujú.
Hay mucha gente que le gustaría seguir como en la parte de academia, entonces siguen haciendo un postdoc que puede durar 1 o 2 años y seguir como por esa fase, luego a volverse profesor asistente, etcétera, hasta de pronto llegar a ser como decir, un PI, un profesor, ¿no?
Ok.
Y un jefe como de laboratorio. Pero hay mucha gente que también después del doctorado dice como listo, sí, me gustó la parte de, digamos, investigación y eso, pero prefiero irme de pronto a la parte de industria. Entonces se van ya a una compañía donde también puedan ayudar, no sé, de parte de inmunología, donde se pueden hacer otro tipo de, de ensayos y eso. Entonces mucha gente también opta o sea, por la otra área que es compañías, la parte como de farmacéutica y eso.
Ok, perfecto, listo. Tenemos dos preguntas. La primera es, ¿podrían estos hallazgos aplicarse a la infección con algún otro parásito?
Pues en el caso, o sea, que digamos el que se vio primero, de pronto hasta el momento se han visto con los relacionados, digamos, con malaria. Ahí hablamos, digamos, de tripanosoma, de toxoplasma, también de pronto de leishmania. Eso se podría mirar, porque lo que yo les mostré al principio de que se vio que por lo menos lo que fueron las proteínas formadoras de poros, lo que fue perforina, granulosina y granzima ayudan y también ayudan a disminuir la infección, ¿no?, de los parásitos intracelulares, como les dije, tripanosoma. En el caso tripanosoma cruzi, que es el que causa Chagas, eso podría ser muy bueno. Digamos en Colombia que también tenemos tantas infecciones. En el caso también de leishmania y, por ejemplo, toxoplasma. Entonces, por el momento, más que todo a nivel intracelular, o sea, con los parásitos intracelulares.
Ok, listo, perfecto. La pregunta, vuelve profesora Ana Lucía a preguntar, ¿hay alguna posibilidad que en la universidad donde tú estás en este momento haya acercamientos para hacer pasantías de estudiantes de pregrado del programa de microbiología?
En estos momentos no estoy segura. O sea, yo sé que acá, por lo menos con los estudiantes de acá, reciben, pero más que todo para maestría. No podría estar 100% segura si de pronto reciben para una pasantía. La pasantía sería más o menos, yo creo que de 6 meses. Entonces, no sé si, no sé la verdad, pero pues podría preguntar, la verdad, porque no, la verdad no conozco, hasta desde los 3 años que llevo acá, no conozco que haya venido un pasante, digamos, del exterior.
Ok, listo, pero
Eso sí, no.
digamos por ejemplo que pues el requisito inicial para eso eventualmente sería que tuvieran inglés, ¿cierto? El nivel de inglés. Pues
Por lo menos,
tú estás
sí,
en la zona francesa de Suiza, pero digamos que si llega una persona que ya tiene un buen nivel de inglés podría defenderse, ¿cierto?
sí, sí, porque acá por lo menos con, o sea, lo que es con mi profesor, o sea, todas las reuniones se hacen en inglés, con mi profesor todo es en inglés, francés digamos que lo utilizo a veces cuando tengo que hablar con alguna de las técnicas o de pronto principalmente con las personas que se encargan de los animales. Hay algunas personas que su inglés no, o sea, no es muy bueno, entonces ahí digamos que sí, pero el resto todo es en inglés, digamos que dentro de la universidad. Entonces, si tienen un nivel de inglés, bueno, y si, o sea, y si hacen, digamos, el IELTS o el TOEFL, entonces no hay ningún problema.
Ok, bueno, listo. Yo creo
Ujú.
que